Hello, and welcome back to another episode of DNA Diaries. Today, we're going to be discussing gene regulation, a microscopic process with macroscopic implications for our bodies. We're going to look at research done on the regulation of the gene for Hemeoxygenase-1, but first, let's take a step back and set the scene. You and me are made of billions of cells that carry out all the different functions we need to survive. There are a lot of things out there that can harm our cells, but luckily, the body has its own built-in mechanisms to prevent some of the more common forms of damage. One threat to our cells are chemicals called reactive oxygen species, or ROS. ROS are byproducts of metabolism, and if too many accumulate, they can cause oxidative stress and damage our cells. We do kind of need our cells, so our body has developed a defense system against ROS, and it comes in the form of an army of proteins called antioxidant enzymes that transform ROS into stable, non-toxic molecules. Hemeoxygenase-1, or HO1, is one of these enzymes. The expression of HO1 can be increased or decreased in response to a cell's exposure to certain chemicals, and this altered expression can be explained by looking at the changes that occur at the level of gene regulation. To examine regulation, we must zoom in on the promoter controlling the transcription of the HO1 gene. This promoter contains two inducible enhancers, E1 and E2. Within each enhancer, there are three antioxidant response elements, or AREs, that proteins called transcription factors can bind to in order to regulate HO1 expression. Many transcription factors that bind ARE sequences in the promoter region of the HO1 gene have been identified, but their regulatory mechanisms are unclear. Today, we're going to be focusing on the results of a study that analyzed the roles of two of these transcription factors in the regulation of ARE-mediated HO1 expression. The journal article we'll be discussing is by Jinsung Park and Hee-Sung Kim. It focuses on the regulatory mechanisms of the transcription factors NRF2 and CJUN in the expression of HO1 in primary rat astrocytes stimulated with the antioxidant tertiary butyrylhydroquinone, or TBHQ. The researchers decided to explore this question using rat astrocytes because these cells regularly express HO1 to protect neurons in the brain from damage. TBHQ was chosen as a cell stimulant because it upregulates HO1 expression, which allows changes in transcription factor activity to be observed. To kick off the experiment, the researchers wanted to know how TBHQ went about increasing the expression of HO1. Remember that promoter diagram we saw at the start of the video? Well, the researchers decided to investigate whether or not TBHQ regulates HO1 expression by affecting the binding of transcription factors to the ARE sequences in the enhancers of this promoter. To do this, they created HO1 reporter plasmids where the regulatory E1 and E2 regions of the HO1 gene are fused to the reporter gene luciferase. They created four reporter constructs, one with E1, one with E2, one with mutated E1, and one with mutated E2. The first two enhancers are normal, or wild type, and the last two have the ARE sites removed so transcription factors cannot bind. In these constructs, the HO1 regulatory regions regulate the expression of luciferase instead of HO1, but the same levels of expression would be produced in the case of either gene. Luciferase is just used instead because it glows from bioluminescence and is easy to visualize when doing research. The researchers took these four reporter plasmids and put them into astrocytes. The first figure shows the full induction in luciferase expression from the E1 constructs, and the second figure shows the E2 constructs. Fold induction is the ratio of experimental expression to the control expression of cells not exposed to TBHQ. The higher the fold induction, the greater the increase in gene expression. When the astrocytes were treated with increasing concentrations of TBHQ, luciferase activity increased in the cells with the two wild-type promoters, as shown by the black bars. But this increase was not observed in cells where the ARE sequences in the promoter were mutated. These results indicate the importance of those ARE sequences and show that TBHQ upregulates HO1 by enhancing promoter activity in an ARE-dependent manner. We now know that TBHQ increases HO1 expression by enhancing promoter activity at the ARE sequences. The next question the researchers asked was, what transcription factors are binding to the ARE sequences in the promoter to cause this TBHQ-mediated upregulation? The transcription factors NRF1, NRF2, CJUN, and CFOS had previously been implicated in ARE binding, so they had a suspicion that these proteins could be involved in the regulation of HO1. To test this, they stimulated astrocytes with TBHQ for 3 hours, then treated the samples with antibodies against each of the four transcription factors separately, which decreased the amount of the targeted transcription factor in that sample. Nuclear extracts from each sample underwent a process called agarose gel electrophoresis, where samples were loaded in wells at the top of the gel before a current was run through it. Smaller proteins and DNA fragments have an easier time migrating through the gel, so they're found on the bottom, while larger proteins and fragments are found on top. After the nuclear extracts were separated by size, an ARE probe was used to identify where the ARE sequences were on the gel. Here's the image they got as a result. The bands represent the ARE probe where the ARE probe is bound on ARE sequences in the gel, and thicker bands indicate a higher concentration of AREs. The dark bands at the bottom represent free ARE with no transcription factors bound, and the two bands towards the top of the gel, shown under each sample, represent the ARE protein complex with transcription factors bound. The ARE protein complex bands for the samples where we lowered levels of NRF2 and c-Jun are the only ones that look lighter than the bands for the control sample where none of our 4 transcription factors of interest were reduced. This suggests that these 2 transcription factors bind ARE in the enhancers, as they are the only transcription factors where removal results in diminished detection of proteins on the ARE sequence. The TBHQ-mediated upregulation of HO1 also decreased when NRF2 and c-Jun were removed, showing that their binding to ARE is involved in enhancing HO1 expression. When combined with what we know about TBHQ upregulating HO1 through ARE sequences, we can now make the conclusion that TBHQ does this by increasing the binding of NRF2 and c-Jun to ARE in the promoter of the HO1 gene. Knowing that NRF2 and c-Jun are the transcription factors involved in enhancing HO1 expression, the next question we can ask is, what specific roles do these transcription factors play in the regulation process? To figure this out, the researchers used small interfering RNA, or siRNA, to selectively remove either NRF2 or c-Jun in astrocytes stimulated with TBHQ to determine what effect this would have on HO1 expression. They compared levels of HO1 protein before and after 6-hour TBHQ treatment for 4 different samples. The white bars represent the initial or basal expression levels of HO1 in the samples, and the gray bars show HO1 expression after the astrocyte is exposed to TBHQ. The control siRNA and no siRNA conditions were the two samples where NRF2 and c-Jun were both present at normal levels, and these samples both showed increases in HO1 expression after TBHQ treatment. When NRF2 was knocked out and only c-Jun was present in the samples, there was a decrease in basal levels of HO1 expression before TBHQ treatment, but the relative increase in HO1 expression after TBHQ treatment stayed constant. On the other hand, when c-Jun was knocked out and only NRF2 was present, basal levels of HO1 expression remained the same, but exposing the cells to TBHQ didn't increase the expression of HO1. These results show that NRF2 and c-Jun play different regulatory roles when controlling the expression of HO1. NRF2 modulates basal expression of HO1, while c-Jun mediates the increase in HO1 expression in response to TBHQ. That's a lot of information to take in, so let's take a moment to summarize some of the main findings. Recall that astrocytes protect surrounding neuronal cells against oxidative stress, and one of the enzymes they use to accomplish this is HO1. This study investigated the regulatory mechanisms of the transcription factors NRF2 and c-Jun in controlling HO1 expression in primary astrocytes treated with TBHQ. TBHQ was found to increase HO1 expression by enhancing HO1 promoter activity in ARE sequences. Through TBHQ increasing the binding of transcription factors NRF2 and c-Jun to these ARE sequences, NRF2 was able to maintain basal levels of HO1 expression, while c-Jun mediated HO1 induction. So why should we care so much about all this anyway? I promise I didn't just talk your ear off about HO1 regulation for nothing. It turns out that HO1 is overexpressed in tumor tissues. Since HO1 prevents cellular damage, when its expression can't be turned off, it prevents the death of mutated tumor cells, promotes tumor growth, and facilitates the spreading of cancer to different parts of the body. The regulation of HO1 expression is of therapeutic interest because if we can understand how this gene is controlled, we may be able to downregulate its expression in order to prevent it from contributing to tumor growth in patients with cancer. Before we wrap up today's episode, we need to address a limitation in this study. It's easy to get excited about results, but we need to remember that this research only investigated the roles of NRF2 and c-Jun in primary astrocytes that were upregulated by TBHQ. The roles of these transcription factors in HO1 expression may be different depending on what types of cells and stimulants are used, so further studies examining the roles of NRF2 and c-Jun in different contexts are needed. Despite these limitations, the insight gained into the regulation of HO1 expression through the research done by Park and Kim goes a long way towards developing therapeutic approaches to combat tumor growth. Stay tuned for more on this gene regulation story. See you next time on the DNA Diaries.